Imagine waking up tomorrow and being told that the last person who will ever die of old age has already been born. Not in some distant science fiction future, but that the child alive right now, somewhere on this planet, will be the final human being to grow frail and surrender to time. What if aging itself, the slow, relentless decay that has claimed every single human who has ever lived, from the earliest Homo sapiens huddled around fires in Africa to the greatest emperors and philosophers and warriors in history, is not an inevitability, but simply a problem that has not yet been solved? Because that is exactly what a growing number of the most brilliant scientists, engineers, and thinkers on Earth believe today. Not poets, not mystics, scientists. And they are not talking about living to 120 or 150. They are talking about something far more radical, the complete and permanent defeat of death itself. This is not a video about living a long life. This is a video about living forever. Let's take a moment to truly sit with what we are discussing. Every single person you love will die. Your parents, your friends, your partner, every child born today, every leader currently shaping the world. Billions of people alive right now are scheduled to cease to exist within a century. We treat this as normal. We treat this as natural. We build entire religions, philosophies, and cultures around the acceptance of it. And yet, what if we are simply the last generation that has to? What if the death of aging is closer than anyone thinks? Over the course of this video, we are going to explore the full landscape of human immortality, from the ancient obsession with eternal life, to the cutting-edge science happening right now in laboratories around the world. We are going to look at what aging actually is at the biological level, and why some of the world's top scientists now believe it can be stopped, slowed, or even reversed. We will examine the technologies that are moving fastest: senolytics, gene editing, AI-driven drug discovery, whole brain emulation, and nanotechnology. We will confront the real philosophical, ethical, and social questions that immortality raises, questions about meaning, overpopulation, inequality, and the nature of identity itself. And we will try to answer the question that everyone watching this video is probably already asking: could people alive today actually live forever? Buckle in. This is going to change how you think about everything. To understand where we are going, we need to understand where we have always been. The desire to defeat death is not new. It is arguably the oldest desire in human history. The Epic of Gilgamesh, written over 4,000 years ago in ancient Mesopotamia, tells the story of a king who watches his best friend die, and is so shattered by the experience that he sets out on a journey to find eternal life. He finds a plant at the bottom of the ocean that supposedly grants immortality, only to have it stolen by a serpent while he sleeps. He returns home empty-handed, mortal. The story ends not with triumph, but with Gilgamesh simply looking at the walls of his city, recognizing that the only immortality available to him is the legacy he leaves behind. The ancient Egyptians built an entire civilization around the denial of death. Their pharaohs were buried in monuments designed to last eternity. They mummified the dead because they believed the body needed to be preserved for the soul's return. They mapped the afterlife in elaborate detail, filling tombs with food and treasures and instructions for navigating the underworld. The ancient Chinese emperors sent expeditions to find the elixir of immortality. Qin Shi Huang, the first emperor of China, who unified the country in 221 BCE, was so obsessed with living forever that he reportedly consumed pills containing mercury, a known poison, believing they were the key to eternal life. They killed him instead. In ancient Greece, the gods were defined precisely by their immortality. They were the athanatoi, the deathless ones. Humans were netoi, the mortal ones. This distinction was not incidental. It was fundamental to what it meant to be human. And yet the Greeks also told the story of Tithonus, a mortal man whom the goddess Eos loved so much that she asked Zeus to grant him eternal life. She forgot to ask for eternal youth. Tithonus lived forever, but grew older and more decrepit without end, eventually shrinking into a cricket, unable to die but also unable to truly live. This story is often invoked as a warning: be careful what you wish for. But notice something. The Greeks were not warning against immortality itself. They were warning against immortality without health. The dream was always eternal youth, not just eternal existence. Across cultures and centuries, human beings have dreamed of the same thing: the fountain of youth, the philosopher's stone, the Hindu concept of Amrita, the Jewish legend of the elixir of life, the Christian promise of resurrection, the Taoist pursuit of Zion, transcendence of mortality. Every civilization that has ever existed has produced a mythology of immortality. And every civilization has also, ultimately, accepted that immortality was beyond human reach. Until now. Because something extraordinary has happened in the last few decades. The people who are now seriously pursuing immortality are not alchemists or mystics or priests. They are molecular biologists, gerontologists, geneticists, neuroscientists, and computer scientists. And rather than searching for magical plants or divine elixirs, they are doing something far more powerful. They are trying to understand, at the most fundamental possible level, what aging actually is, and then trying to fix it. Let's talk about what aging really is. Because most people have a vague sense that the body just kind of wears out over time, like an old car. But that metaphor is not quite right, and understanding why it's wrong is crucial to understanding why some scientists think it can be defeated. Your body is not a machine in the sense that it is made of fixed parts that simply erode. Your body is a dynamic, self-repairing system of extraordinary complexity. Right now, as you watch this video, your cells are constantly dividing, replacing, repairing. Your skin replaces itself roughly every few weeks. Your red blood cells are replaced every 4 months. The cells lining your gut turn over every few days. If your body is so good at repairing itself, why do you age at all? The answer lies in a series of interconnected biological processes that scientists have been unraveling over the past few decades. In 2013, a group of leading researchers published a landmark paper in the journal Cell, identifying what they called the 9 hallmarks of aging. These are the fundamental biological mechanisms that drive the aging process. Understanding these hallmarks is understanding what we would need to fix to stop aging. The first hallmark is genomic instability. Every time a cell divides, it has to copy all 6 billion base pairs of your DNA. Over decades, errors accumulate. Some of these errors are repaired. Many are not. Damaged DNA leads to dysfunctional cells, and dysfunctional cells lead to everything from cancer to organ failure. The second hallmark is telomere attrition. Telomeres are protective caps at the ends of your chromosomes, a bit like the plastic tips on the ends of shoelaces. Every time a cell divides, the telomeres get a little shorter. When they get too short, the cell can no longer divide. It either dies or becomes what is called a senescent cell, a zombie cell that stops functioning properly but does not die. The third hallmark is epigenetic alterations. Your genes are not a fixed program. They are regulated by epigenetic marks, chemical tags that tell genes when to turn on and off. These marks change over time in ways that are dysregulated and harmful. Genes that should be on get turned off. Genes that should be off get turned on. This contributes enormously to the aging process and to age-related disease. The fourth hallmark is loss of proteostases. Your cells contain thousands of different proteins, each with a specific shape that determines its function. Over time, proteins misfold and aggregate. Your cells have systems for clearing out damaged proteins, but these systems decline with age. Misfolded protein aggregates are the hallmark of diseases like Alzheimer's and Parkinson's, and they accumulate throughout the body as we age. The fifth hallmark is deregulated nutrient sensing. Your cells have sophisticated systems for detecting how much food is available and adjusting their behavior accordingly. Pathways like mTOR, insulin signaling, and sirtuins regulate how cells grow, divide, and repair themselves based on nutritional signals. As we age, these pathways become dysregulated in ways that accelerate aging. The sixth hallmark is mitochondrial dysfunction. Mitochondria are the power plants of your cells, producing the energy currency that drives everything your body does. Over time, mitochondria accumulate damage and become less efficient. Dysfunctional mitochondria not only produce less energy, but also generate more harmful reactive oxygen species, free radicals that cause further cellular damage. The seventh hallmark is cellular senescence. We mentioned senescent cells earlier. These are cells that have stopped dividing but haven't died. They accumulate throughout the body with age, and rather than simply being inert, they actively secrete a cocktail of inflammatory chemicals called the senescence-associated secretory phenotype, or SASP. This chronic, low-grade inflammation, sometimes called inflammaging, drives a huge amount of age-related disease and dysfunction. The eighth hallmark is stem cell exhaustion. Throughout your body, stem cells serve as reservoirs for tissue repair. When you injure your muscle, stem cells divide to replace the damaged fibers. When you lose cells in your gut or your skin, stem cells replenish them. But as you age, your stem cell populations decline, and the stem cells that remain become less capable. This is why wounds heal more slowly as you get older, and why the body becomes progressively less able to maintain and repair itself. The ninth hallmark is altered intercellular communication. Cells don't function in isolation. They are constantly sending and receiving signals through hormones, growth factors, cytokines, and direct contact. As we age, this communication becomes increasingly dysregulated. Chronic inflammation disrupts signaling. Hormonal changes alter how cells communicate. The result is a progressive breakdown of the coordinated tissue function that health depends on. Since that landmark 2013 paper, the field has continued to evolve. Some researchers have proposed additional hallmarks that deserve attention. Chronic inflammation, often called inflammaging, is increasingly recognized as a master driver of virtually all age-related pathology. Dysbiosis, or changes in the gut microbiome, have been linked to systemic aging in ways that scientists are only beginning to understand. And altered mechanical properties of tissues, the stiffening of arteries, the cross-linking of collagen, the loss of tissue elasticity, represent a physical dimension of aging that operates somewhat independently of the cellular and molecular processes described above. But what makes all of this both fascinating and hopeful is that these 9 hallmarks are not random, unpredictable events. They are specific, identifiable biological processes. And if they are specific biological processes, then they are, at least in principle, addressable. You can imagine interventions that target each one of these hallmarks, and that is exactly what scientists are now doing. Before we get to the most dramatic recent experiments, we need to spend a moment on the deep historical backdrop of longevity science, because the clues were always there, embedded in biology, long before anyone thought to look for them seriously. The first and perhaps most important clue came from a simple dietary observation. In 1935, a Cornell University biochemist named Clive McKay discovered something startling. Laboratory rats that were given 30 to 40% fewer calories than they would naturally eat, while still receiving adequate nutrition, lived dramatically longer than normally fed rats. This was not a trivial extension. The calorie-restricted rats lived up to 50% longer than controls. They also stayed healthier for longer, developing fewer tumors, maintaining better organ function, showing fewer signs of age-related decline. This observation was replicated over the following decades in species after species. Caloric restriction extended lifespan in yeast, in worms, in flies, in fish, in rodents. In every organism studied, eating less meant living longer. The consistency of this finding across the entire tree of life suggested that the mechanisms involved were ancient, evolutionarily conserved, and fundamental to how living systems work. The biological explanation took decades to emerge. What scientists eventually discovered is that caloric restriction activates a set of cellular stress responses that shift the organism away from growth and reproduction and toward maintenance and repair. When food is scarce, it makes evolutionary sense for the organism to hunker down, to stop growing, to pause reproduction, to ramp up cellular repair mechanisms, to become as efficient as possible, and to wait for better times. This maintenance mode turns out to be associated with dramatically slower aging. The key molecular pathways involved include mTOR, mechanistic target of rapamycin, which acts as a master sensor of nutrient availability and a master regulator of cellular growth and metabolism. When food is plentiful, mTOR is active, the cell grows and divides, and cellular maintenance takes a back seat. When food is scarce, mTOR is inhibited, growth slows, and the cell shifts resources toward maintenance and repair. Critically, this maintenance mode involves upregulation of autophagy, the cellular process of breaking down and recycling damaged proteins and organelles. Autophagy is, in essence, the cell's housekeeping system, and stimulating it has consistently been shown to extend lifespan in experimental organisms. This understanding led to one of the most important drug discoveries in aging science, rapamycin, an immunosuppressant drug originally isolated from a soil bacterium found on Easter Island. Rapamycin inhibits mTOR directly. When aging researchers began testing it in mice, the results were extraordinary. Even when rapamycin was administered only to middle-aged mice, the equivalent of starting treatment in a 60-year-old human, it extended their remaining lifespan by approximately 30%. This was the first drug shown to extend the lifespan of mammals when given in middle age, and it created enormous excitement in the field. Rapamycin is not a perfect drug. It has immunosuppressive effects that limit its use at high doses, but researchers are exploring lower doses and intermittent dosing schedules that might capture the longevity benefits while minimizing side effects. Some physicians are already prescribing it off-label for longevity in relatively healthy patients. The data from human longevity trials will take time to accumulate, but the animal data is remarkably consistent. Another approach that emerged from the caloric restriction research is intermittent fasting. Rather than continuously restricting calories, intermittent fasting cycles between periods of eating and periods of fasting. The biological rationale is similar. Periodic fasting activates the same maintenance and repair pathways as caloric restriction. Clinical data suggests that intermittent fasting has beneficial effects on metabolic health, inflammation, and various biomarkers associated with aging. Whether it extends human lifespan remains to be demonstrated, but the biological basis for believing it might is solid. The worm C. elegans has been particularly important in aging research, precisely because it has a lifespan of only about 3 weeks, making it possible to study aging and longevity in real time. In the 1990s, Cynthia Kenyon at the University of California, San Francisco made a discovery that transformed the field. She found that a single mutation in a gene called DAF2, which encodes a receptor for a hormone similar to insulin, doubled the lifespan of C. elegans. The worms with the mutation were not just alive longer, they were healthier and more active for a greater fraction of their extended lives. This was the first demonstration that a single genetic change could dramatically extend the lifespan of any organism, and it shattered the prevailing assumption that aging was too complex to be altered by simple interventions. Kenyon's discovery opened a floodgate. Researchers identified hundreds of genes that affect lifespan in C. elegans, many of them conserved in mammals. They showed that growth hormone pathways, similar to the insulin signaling pathway Kenyon had identified, were associated with longevity in mice and in humans. Some of the longest-lived human populations were found to have unusual variants in these same pathways. The connection between the humble worm and human aging was real. Similar work was done in Drosophila melanogaster, the fruit fly, and in yeast. In every model organism, scientists found that aging was genetically modifiable, often dramatically. Doubling, tripling, even quintupling the lifespan of simple organisms became routine achievements in aging labs. The organisms themselves provided proof of principle. The machinery of aging is not fixed. It can be tuned. Let's talk about the experiments that first made serious scientists genuinely believe that aging could be reversed. One of the most stunning experiments in the history of aging research is a procedure called heterochronic parabiosis. The name sounds exotic, but the concept is simple and disturbing. Scientists take an old mouse and a young mouse and surgically join their circulatory systems so that they share the same blood. Within weeks, the old mouse begins to show signs of rejuvenation. Its muscle strength improves. Its brain function improves. The regenerative capacity of its tissues increases. Meanwhile, the young mouse begins to age more rapidly. This experiment, which was actually first performed in the 1950s but was dramatically refined and expanded in the 2000s by researchers at Stanford, Harvard, and other institutions, suggests something extraordinary. There are factors in young blood that have the ability to reverse age-related decline, and there are factors in old blood that actively suppress repair and rejuvenation. Researchers like Amy Wagers at Harvard have worked to identify these factors. One important candidate is a protein called GDF11, which appeared to reverse cardiac aging in older mice when infused from young animals. The research on GDF11 turned out to be more complicated than initially appeared and became somewhat controversial, but the broader principle, that circulating factors regulate the pace of aging, has been confirmed repeatedly. Other researchers identified a factor called VEGF and another called oxytocin as being involved in age-related changes. Most dramatically, a research group identified that a factor called TGF-beta was elevated in old blood and actively suppressed the regenerative capacity of muscle and brain stem cells. When this factor was blocked in old animals, their regenerative capacity was partially restored, even without any young blood being introduced. Then came research on a protein called clotho, named after one of the Greek fates who spun the thread of life. Clotho is a protein that declines dramatically with age. When clotho levels are artificially elevated in old mice, they live dramatically longer than control mice. When the Klotho gene is knocked out in young mice, they age extremely rapidly and die young. The discovery of Klotho suggested that there might be specific molecular regulators of the aging rate, and that manipulating those regulators could have profound effects on lifespan. Perhaps the most dramatic experimental results in recent years came from the field of epigenetic reprogramming. We mentioned earlier that one of the hallmarks of aging is epigenetic alterations, changes in how genes are expressed. In 2006, the Japanese scientist Shinya Yamanaka discovered that you could take any cell from an adult body and, by introducing just 4 proteins, now called Yamanaka factors, reset its epigenome to an embryonic state. Adult cells were literally reprogrammed into induced pluripotent stem cells, capable of becoming any tissue in the body. Yamanaka won the Nobel Prize for this discovery in 2012. The obvious next question was, what if you applied the Yamanaka factors not fully, not enough to completely reprogram a cell, but just partially, just enough to reset some of the epigenetic aging without differentiating the cell back into a stem cell? In 2016, scientists in Juan Carlos Ispasua Belmont's lab at the Salk Institute in California tried exactly this in a mouse model of progeria, a disease of accelerated aging. They delivered the Yamanaka factors intermittently and showed that it reversed many signs of aging in the mice and extended their lifespan dramatically. In 2020, the same lab published a study showing that partial epigenetic reprogramming could reverse age-related vision loss in old mice and even restore vision in mice with glaucoma-like damage. The implications of this are staggering. The idea is that aging is not a one-way ratchet. The epigenetic information that defines a young, healthy cell is still present in an old cell. It has just been buried under decades of accumulated noise and dysregulation. If you can reset this epigenetic program, you can restore youthful function to aged cells without erasing their identity. David Sinclair at Harvard University, one of the most prominent aging researchers in the world and the author of the book Lifespan, argues that this is indeed what is happening. He calls this the information theory of aging, the idea that aging is fundamentally a loss of information, specifically epigenetic information, and that aging could be reversed by restoring this information. Sinclair's lab has been at the forefront of research into sirtuins, a family of proteins that regulate gene expression and have been linked to longevity in organisms from yeast to mice to worms. It was research in Sinclair's lab and others that sparked intense interest in molecules like resveratrol and NAD+ precursors like NMN and NR, which activate sirtuin pathways. While the translation of these findings from mice to humans has proven complicated, as is almost always the case in medicine, the underlying biology has motivated an enormous field of research. Meanwhile, an entirely different approach to life extension was gaining momentum. Senolytics. Remember senescent cells, those zombie cells that stop dividing but refuse to die, instead pumping out inflammatory chemicals. By the 2010, researchers had developed drugs capable of selectively killing these cells, drugs called senolytics. The first major proof of concept came from a lab at the Mayo Clinic, led by James Kirkland and Jan van Dusen. They showed that clearing senescent cells from mice using a genetic trick dramatically extended the healthy lifespan of the animals. Then they showed that drugs, specifically a combination of dasatinib and quercetin, could achieve similar effects. The results were remarkable. Old mice that were treated with senolytics showed improvements in physical function, organ health, and even cognitive performance. They looked and acted younger. And crucially, they lived longer, not just longer in the sense of a few extra weeks, but significantly longer, with healthier final years. Senolytic drugs are now in human clinical trials. Companies like Unity Biotechnology and Siwa Therapeutics are pursuing senolytic approaches for everything from eye disease to arthritis to pulmonary fibrosis. The trials are still in early stages, and translating results from mice to humans is notoriously difficult. But the biological rationale is sound, the animal data is compelling, and the scientific community takes the approach seriously. All of this research on aging mechanisms, on epigenetic reprogramming, on senolytics, on blood factors, it is converging on a single remarkable possibility. Aging is not an immutable law of nature. It is a biological process, and biological processes can be engineered. Now let's talk about the people who are putting serious money behind this idea, because nothing signals that mainstream science has accepted a possibility like billionaires and major institutions throwing resources at it. The year is 2013. Google, one of the most powerful technology companies in the world, a company that reshaped the global flow of information, that knows more about you than your closest friends, decides that its next great problem to solve is death. The company they created to pursue this goal is called Calico, which stands for California Life Company. They brought in renowned biologist Cynthia Kenyon, whose research on the genetics of aging in the worm C. elegans had shown that single gene mutations could double its lifespan. They invested hundreds of millions of dollars. They partnered with AbbVie. The mission statement was almost laughably ambitious: to understand the biology that controls lifespan, and to devise interventions that enable people to lead longer and healthier lives. Around the same time, Jeff Bezos was quietly investing in a company called Unity Biotechnology, the senolytic drug company we mentioned earlier. Peter Thiel, the contrarian billionaire investor, made no secret of his interest in life extension. He took human growth hormone and advocated publicly for the possibility of dramatically extended lifespans. He funded the Sense Research Foundation, founded by the British biogerontologist Aubrey de Grey. Aubrey de Grey deserves special attention because for many years he was the most visible and most controversial figure in the longevity field. De Grey is a software engineer turned biomedical gerontologist, which is an unusual background, but it gave him an outsider's willingness to challenge the field's assumptions. His thesis, which he developed in the early 2000s, was called Sense: Strategies for Engineered Negligible Senescence. It argued that aging was fundamentally an engineering problem, not a medical one. Instead of treating age-related diseases one by one, you should target the underlying damage that causes them all. The 7 types of damage he identified overlapped significantly with the later hallmarks of aging framework. He argued that all of these types of damage were, in principle, repairable using known or foreseeable technologies. When de Grey first proposed Sense, he was widely dismissed by mainstream gerontologists. Technology Review magazine held an unusual contest, offering money to anyone who could prove that Sense was so wrong that it was unworthy of learned debate. The judges ultimately decided that while Sense was speculative, it could not be dismissed in this way. Gradually, over the following 2 decades, mainstream science moved significantly in de Grey's direction, not necessarily accepting all of his specific proposals, but accepting the broader premise that aging is something that can be engineered. The real explosion of investment and scientific activity came in the 2020s. OpenAI founder Sam Altman invested $180 million into a company called Retro Biosciences, with the explicit goal of adding 10 years to the healthy human lifespan. Larry Ellison, the billionaire co-founder of Oracle, famously said he found the prospect of death incomprehensible and has donated hundreds of millions of dollars to aging research. Jeff Bezos quietly co-funded a company called Altos Labs with Yuri Milner, a Russian-Israeli billionaire technology investor, raising over $3 billion. Altos recruited some of the biggest names in aging biology, including Shinya Yamanaka himself, the discoverer of the reprogramming factors, and set them to work on partial epigenetic reprogramming in the pursuit of biological rejuvenation. Meanwhile, the NIH launched a major program focused on aging. Academic institutions created dedicated aging research centers. The whole tone of the scientific conversation shifted. Aging was no longer a background condition of life that medicine managed around. It was becoming a target. And then there is the company that has perhaps generated more attention in the longevity space than any other in recent years. Hevolution Foundation in Saudi Arabia, funded by the Saudi government, with $1 billion per year allocated to aging research. This is an extraordinary sum, more than the entire NIH budget for aging research. And it signals something important. The largest, most powerful institutions in the world are now treating the defeat of aging as a serious, fundable goal. One of the most exciting recent developments in longevity science is the study of people who have already beaten the odds, supercentenarians and the inhabitants of so-called blue zones. Supercentenarians are people who have lived past 110 years. There are only about 150 of them alive at any given time, scattered across the globe. What makes them remarkable is not just their age but their health. Many supercentenarians remain cognitively sharp and physically active well into their second century. They delay the onset of age-related diseases dramatically compared to the general population. When researchers study their genomes, they find unusual variants in immune system genes, inflammation-related genes, and longevity pathways like APO. Their microbiomes tend to be unusually diverse and rich in certain beneficial bacterial species. Their telomeres tend to be longer than age-matched controls. Studying supercentenarians is difficult because there are so few of them, and many are reluctant to participate in research. But the data that exists suggests that extreme longevity is not simply the result of avoiding death from specific diseases. These individuals seem to have constitutively lower rates of the biological aging processes we described earlier: lower inflammation, better DNA repair, more resilient stem cell populations. Understanding what makes them different is a major research priority, because it could point toward interventions that mimic their advantages in everyone else. The blue zones concept, popularized by the journalist and explorer Dan Buettner, identifies 5 regions of the world where people live significantly longer and healthier lives than average: Okinawa in Japan, Sardinia in Italy, Nicoya in Costa Rica, Icaria in Greece, and Loma Linda in California. Careful study of these populations has identified several common factors: high physical activity as part of daily life rather than deliberate exercise, predominantly plant-based diets, low levels of chronic stress, strong social connections, a sense of purpose, and moderate alcohol consumption in some groups. These are all things that map onto the biological hallmarks of aging: reducing inflammation, maintaining cellular repair, supporting mitochondrial function, preserving social engagement that buffers psychological stress. The blue zones research has been somewhat controversial, with some researchers questioning whether the longevity statistics in these populations are as robust as claimed. But the broad message, That lifestyle factors have profound effects on healthy longevity is consistent with an enormous body of research. And crucially, understanding why lifestyle factors work at the biological level can point toward drugs and interventions that replicate those effects for people who cannot achieve them through lifestyle alone. The gut microbiome has emerged as an unexpectedly important player in aging. The trillions of bacteria, archaea, fungi, and viruses that inhabit your intestines are not passive passengers. They interact dynamically with your immune system, your metabolism, your nervous system, and virtually every organ in your body. They produce metabolites that influence inflammation, energy metabolism, and even mood and cognition. And they change dramatically with age, generally becoming less diverse and less populated with beneficial species. Research has shown that transferring gut bacteria from young mice to old mice has rejuvenating effects, improving gut barrier function, reducing systemic inflammation, and even improving brain health. Conversely, transferring gut bacteria from old mice to young mice can accelerate aging-like changes. This suggests that the microbiome is not just a passive reflection of the body's aging, but an active participant in it. Specific microbial species and the metabolites they produce are being intensively studied. Akkermansia muciniphila, a bacterium that maintains the integrity of the gut lining, declines dramatically with age and is associated with metabolic health. Supplementing it in mice and in early human trials shows promising results. Other researchers are focused on short-chain fatty acids like butyrate, produced by beneficial gut bacteria, which have anti-inflammatory and epigenetically active properties. The emerging field of longevity-focused microbiome intervention is still young, but the potential is significant. The immune system deserves special attention in any discussion of aging. The immune system does not simply decline uniformly with age, a process called immunosenescence. Rather, it becomes dysregulated in specific ways. The adaptive immune system, the branch that generates specific responses to new threats, becomes less capable of responding to novel challenges. This is why older people are more vulnerable to new infections, and why vaccines tend to be less effective in the elderly. At the same time, the innate immune system, the branch that generates nonspecific inflammatory responses, becomes hyperactive. This produces the chronic background inflammation called inflammaging that drives so much age-related pathology. The thymus, the organ that produces T cells, the specialized immune cells of the adaptive immune system, begins to shrink and fill with fat starting in early adulthood. By middle age, most of its immune-generating capacity is gone. Researchers are exploring ways to regenerate thymic tissue and restore the production of fresh T cells. In 2019, a clinical trial led by Gregory Fahy at Intervene Immune showed that a combination of growth hormone, metformin, and DHE appeared to reverse biological aging by about 2.5 years in the participants. And the most prominent effect was regeneration of thymic tissue. The study was small and preliminary, but the thymic regeneration finding was remarkable and has motivated further research. Understanding and reversing immune aging is particularly important because the immune system's role is not limited to fighting infection. The immune system also performs surveillance for cancer cells, eliminating them before they become tumors. It clears senescent cells. It oversees tissue repair. When it becomes dysregulated with age, all of these functions suffer simultaneously, contributing to multiple aspects of aging pathology. Now, all of this science is advancing rapidly, but it is advancing in the context of a fundamental challenge that we should discuss directly: the problem of complexity. The human body is not a collection of independent systems. It is an integrated network in which every part affects every other part. Intervening in one pathway inevitably has effects on others. Drugs that extend lifespan in mice routinely show different effects in humans, sometimes because the biology differs, sometimes because mice live in controlled environments that do not reflect the complex exposures of human life, and sometimes because the human aging process involves interactions that simply do not exist in short-lived animals. The field has experienced many cases where a promising intervention in mice failed to translate. Managing this complexity, understanding the full network of interactions before intervening, is one of the central challenges of aging medicine. This is precisely where artificial intelligence enters the picture, and where the pace of progress is about to dramatically accelerate. One of the biggest bottlenecks in any field of medical research is the sheer complexity of the biological systems involved. The human body contains roughly 37 trillion cells. Each cell contains billions of molecules interacting with each other in ways that are almost incomprehensibly complex. Understanding how all of these interactions give rise to health and disease, and finding drugs or other interventions that safely shift the system from a diseased state to a healthy one, is a problem of enormous computational and experimental difficulty. Drug discovery has historically taken over a decade and cost over $1 billion per successful new drug, and the failure rate has been staggering. Artificial intelligence is changing this. In 2020, DeepMind's AlphaFold artificial intelligence system solved a problem that had stumped biochemists for 50 years, the protein folding problem. Proteins are the molecular machines of life, and their function is determined by their three-dimensional structure. But predicting the structure of a protein from its amino acid sequence had proven extraordinarily difficult. AlphaFold could do it with stunning accuracy and at unprecedented speed. Almost overnight, the three-dimensional structures of virtually every protein in the human body became available. This was not just a scientific achievement, it was a practical tool. Knowing the structure of proteins involved in aging processes, understanding exactly how they fold, where their active sites are, how they interact with other molecules, is tremendously valuable for designing drugs that target them. AlphaFold and its successors are being used right now by aging researchers to identify and design interventions that would have taken decades to develop using traditional approaches. More broadly, AI systems are being trained on the vast datasets generated by biological research. Genomic data, proteomic data, metabolomic data, clinical data from millions of patients, and are being used to identify patterns, generate hypotheses, and even design novel therapeutic molecules. Companies like In Silico Medicine have used AI to identify a new drug candidate for fibrosis in a fraction of the time that traditional drug discovery would have required, and have advanced that candidate into human clinical trials. Recursion Pharmaceuticals has built a system that runs millions of biological experiments automatically, using AI to interpret the results and guide the next round of experiments. For aging specifically, AI is being used to develop what are called biological age clocks. These are algorithms that take measurements of biological markers, patterns in DNA methylation, levels of various proteins in the blood, gene expression profiles, microbiome composition, and use them to estimate not your chronological age, but your biological age. How old is your body actually functioning? Some of the most striking findings from these clocks are that biological and chronological age can diverge significantly. Some 70-year-olds have the biological age of a 50-year-old. Some 40-year-olds have the biological age of 60. And crucially, these clocks can measure whether interventions, drugs, lifestyle changes, epigenetic reprogramming, actually reduce biological age. This is hugely important because it solves a fundamental problem with aging research. To test whether a treatment extends human lifespan, you would traditionally have to wait for people to die, which might take decades. But if you can measure biological age accurately, you can test whether an intervention reduces biological age in a much shorter time frame, and use that as a proxy for whether it would extend lifespan. The clocks are not perfect, but they are getting better rapidly, and they are transforming the pace at which aging interventions can be tested. One of the most compelling demonstrations of what AI and biological aging clocks can do together comes from a company called Giro, which has used AI analysis of wearable device data, simple step count and activity data, to show that the pace of biological aging can be inferred from how quickly the body's physiological parameters fluctuate and recover from perturbations. The healthier and younger the body, the more rapidly it returns to equilibrium after being perturbed by exercise or stress or illness. The older the body, the more slowly it recovers. This is a remarkably elegant insight that connects aging theory to something measurable with consumer technology. Other AI systems are being used to analyze existing drugs, the millions of compounds that have already been approved by the FDA for other purposes, and identify which ones might have effects on aging pathways. Metformin, a common diabetes drug, has attracted enormous attention because epidemiological data suggests that diabetics who take it have lower rates of cancer, cardiovascular disease, and other age-related conditions than diabetics who don't, and even comparable rates of these diseases to non-diabetics, despite having diabetes. The TAME trial, targeting aging with metformin, is the first clinical trial explicitly designed to test whether a drug can extend the healthy human lifespan, and it is treating aging itself, rather than any specific disease, as the indication. If this trial succeeds, it will represent a landmark moment, the regulatory and scientific establishment formally acknowledging that aging is a treatable condition. But let's go further, because everything we have discussed so far, senolytics, epigenetic reprogramming, AI drug discovery, metformin, these are, in a sense, incremental approaches. They are trying to slow aging or even partially reverse it, but they are working within the constraints of biology as it currently exists. What about more radical approaches? What about gene editing? The discovery of CRISPR-Cas9, a precision gene editing tool derived from a bacterial immune system, was one of the most important scientific events of the 21st century. Its inventors, Jennifer Doudna and Emmanuel Charpentier, received the Nobel Prize in Chemistry in 2020. CRISPR allows scientists to target specific sequences of DNA in a living cell and cut, edit, or replace them with extraordinary precision. It is, in essence, a search and replace function for the genome. For aging research, CRISPR opens up an enormous range of possibilities. You could potentially correct the genomic damage that accumulates over a lifetime. You could knock in protective variants of genes, like the APOE4 variant that is associated with dramatically reduced risk of Alzheimer's disease, or the rare PCSK9 mutations that protect against heart disease. You could activate genes known to be associated with extreme longevity, like the genes found in supercentenarians, people who live past 110, which researchers are actively studying. Going even further, you could imagine adding entirely novel genes, sequences that do not exist in the human germline, that confer additional protective or longevity-promoting functions. This is the territory of genetic enhancement, and it is deeply controversial, but the biology is becoming feasible even as the ethics lag behind. The story of He Jiankui illustrates both the potential and the peril of this territory. In 2018, a Chinese scientist announced that he had used CRISPR to edit the embryos of two twin girls, creating the world's first gene-edited humans. The edit was to the CCR5 gene, intended to confer resistance to HIV infection. He was widely condemned by the scientific community, not because gene editing is inherently wrong, but because the edit was unnecessary for these particular children. The full consequences were unknown, and the process bypassed ethical review. He Jiankui was imprisoned by Chinese authorities, but the milestone stood. Human germline editing had been done. The genie was out of the bottle. The scientific and regulatory debate about gene editing continues. But what is clear is that as CRISPR and its successors, base editors, prime editors, and other tools with even greater precision, become more refined, the ability to make meaningful interventions in the genetic determinants of aging will grow rapidly. Now let's venture into territory that is even more radical, because we have been talking primarily about extending the lifespan of the biological body. But there is another approach to immortality that does not try to maintain the body at all. It tries to preserve the self. This approach goes by various names: mind uploading, whole brain emulation, digital consciousness transfer. The basic idea is that the mind, the subjective experience, the memories, the personality, everything that makes you you, is the result of information processing in the brain. And if that is true, then in principle, the information could be extracted from the biological substrate and instantiated in a different substrate. A computer, a simulation, some other medium entirely. This idea is not new. It has been a staple of science fiction for decades, but it is increasingly being taken seriously by scientists and philosophers. The key question is whether it is technically feasible, and the answer depends on how you think about consciousness. The most serious scientific project working toward mind uploading is called the Human Connectome Project, an initiative funded by the NIH to map the complete wiring diagram of the human brain, the connectome. The brain contains approximately 86 billion neurons, each connected to thousands of others through synapses. The total number of synaptic connections is estimated at over 100 trillion. Mapping this entire network at the resolution of individual synapses would require extraordinary technologies, and the human brain is currently far beyond our mapping capabilities. But progress is being made. In 2023, researchers at Google and Harvard produced the most detailed map of brain tissue ever created, mapping a 1 cubic millimeter sample of human cortex in extraordinary detail, identifying 57,000 cells, 150 million synaptic connections, and producing a dataset of 1.4 petabytes. 1 cubic millimeter is a tiny fraction of the brain, but the achievement demonstrated that the underlying technology, electron microscopy combined with AI image analysis, can work at the scale required. The question of whether uploading the connectome would upload you, whether it would preserve consciousness or merely create a copy, is one of the deepest philosophical questions in the field. If you were to upload your connectome, and then both the upload and your biological body existed simultaneously, which one would be you? This is not a trivial question, and different frameworks for thinking about personal identity give different answers. Derek Parfit, the philosopher who thought more deeply about personal identity than perhaps anyone in the 20th century, argued that questions about whether the upload is really you rest on confused notions of what personal identity means. Parfit suggested that what matters is not the continuity of some metaphysical self, but the continuity of psychological connections, memories, personality, values, intentions. If the upload has all of those things, then from the practical standpoint of what matters to you about your future self, the upload is you. But many people find this answer deeply unsatisfying because it seems to ignore something crucial: the subjective feeling of being you, the first-person experience of consciousness. This gets into what philosophers call the hard problem of consciousness, the question of why physical processes in the brain give rise to subjective experience at all. We do not have a satisfying scientific theory of consciousness. We do not know whether a sufficiently faithful simulation of a brain would be conscious, or would merely behave as if it were conscious. Until we understand consciousness better, we cannot know whether mind uploading would be a form of immortality, or simply a very elaborate form of copying. These questions are not merely academic. Companies like Nectome are working on brain preservation methods that they claim could preserve the connectome for future upload. Their pitch is explicitly about immortality. They argue that with sufficiently detailed preservation of brain structure, future technology might be able to recover the information encoded in that structure and reconstitute the person. The catch is that their current preservation method requires that the person be euthanized first, raising obvious legal and ethical complications. Most of the mainstream science community is skeptical that brain preservation at the connectome level can capture everything relevant to conscious experience. The brain is not simply a static wiring diagram. It depends on dynamic electrical and chemical activity, on the exact distribution of proteins and neurotransmitters in each synapse, on the state of gene expression in each cell. Whether these factors can be captured in a preserved tissue sample is unclear, but this is all in the context of current technology. The mind uploading scenario is not necessarily one that requires near-term technology. It is a long-term possibility, dependent on advances in neuroscience, AI, and brain imaging that may take many decades. The question is whether those advances will come in time for anyone alive today. There is one more technology we should discuss before turning to the social and philosophical questions: nanotechnology. Richard Feynman first proposed the idea of manipulating matter at the atomic scale in his famous 1959 lecture, "There's Plenty of Room at the Bottom." In the 1980s and 1990s, the futurist Eric Drexler elaborated this idea into a vision of molecular machines, nanoscale robots capable of performing precise physical and chemical operations at the molecular level. Drexler imagined medical nanorobots that could travel through the bloodstream, identify and repair damaged cells, clear out atherosclerotic plaques, kill cancer cells with precision, repair DNA damage, clear misfolded proteins, and perform essentially all of the maintenance that would be required to keep a body indefinitely young. This vision remains largely speculative, but it is not scientifically impossible. Progress in nanotechnology has been significant. DNA nanotechnology, using synthetic DNA strands as a building material for nanoscale structures, has produced remarkable results. Researchers have built DNA origami structures of extraordinary complexity, drug delivery systems that can navigate the body and release their payload only in specific environments, and rudimentary molecular machines that can perform simple operations. In 2023 and 2024, researchers demonstrated increasingly sophisticated drug delivery nanoparticles that could distinguish cancer cells from healthy cells and release therapeutic payloads only inside tumors. These are primitive compared to Drexler's vision of autonomous molecular robots, but they represent real progress in the direction of nanoscale medicine. The timeline for truly transformative medical nanotechnology is deeply uncertain. Futurists like Ray Kurzweil have predicted that nanobot medicine capable of radical life extension will be available by the 2030s or 2040. Mainstream scientists tend to be considerably more conservative, but the trajectory of the technology is clear, even if the pace is disputed. Kurzweil himself is one of the most influential figures in thinking about human immortality, and his framework for understanding technological progress, the law of accelerating returns, deserves mention. Kurzweil argues that technological progress is not linear but exponential. Each generation of technology builds on the last, and this compounding produces acceleration. Information technology has followed this curve reliably for decades. Computing power, storage capacity, network bandwidth have all grown exponentially, while costs have fallen. Kurzweil argues that biology is becoming an information technology, and will therefore follow the same exponential curve. If he is right, then the pace of progress in longevity science will be far greater in the next 20 years than in the previous 20, just as the pace of progress in AI was far greater in the 2010 than in the 1990s. This is the basis for what Kurzweil calls the longevity escape velocity, the idea that if progress in life extension exceeds the rate at which people age, then for every year a person remains alive, science extends their expected lifespan by more than a year. People who reach longevity escape velocity effectively live forever, not because a single silver bullet has been found, but because an ongoing series of incrementally improving treatments keep them one step ahead of aging and death. Kurzweil himself takes over 100 supplements a day, exercises obsessively, and monitors his health with elaborate testing protocols in an attempt to stay healthy long enough for the technology to catch up. He has predicted that longevity escape velocity will be reached around 2030. Most scientists think that timeline is wildly optimistic, but the concept itself, that the relevant threshold to cross is not immortality achieved in a single step, but the rate of progress in life extension, is intellectually compelling. Now we need to turn to a question that has been looming over this entire conversation. Should we actually want this? This is not an idle philosophical question. It is the question that will determine whether and how humanity pursues immortality. And the answers are not simple. The most common objection to immortality is the one about meaning. The argument goes like this: death is what gives life meaning. Because our time is finite, we are forced to choose what to value. The knowledge that this particular sunset, this particular conversation, this particular moment of joy with the people we love, that these things will not last forever is precisely what makes them precious. Remove mortality and you remove the urgency, the poignancy, the weight that makes life feel significant. As the philosopher Bernard Williams argued, an immortal life would eventually become tedious and meaningless, a life without completion, without the shape that death gives to a human story. This is a serious argument, and it is made by serious people, but it has several significant weaknesses. First, it assumes that meaning is derived from the scarcity of time, rather than from the content of experience. But many of the things that give life meaning, love, curiosity, creation, connection, the pursuit of understanding, seem like they could continue to be meaningful indefinitely, not despite being permanent, but because they can be infinitely deepened and varied. A musician does not exhaust the meaningful possibilities of music in a human lifetime. A scientist does not exhaust the questions about the universe. There seems to be no obvious reason why meaning would be exhausted simply because time is abundant. Second, the argument conflates mortality, the certainty of eventual death, with finitude in a particular experience. We already have experiences of finite duration. A meal ends, a concert ends, a friendship might end, a period of life ends. The ending of a particular thing is not the same as the ending of all things, and the meaning of particular experiences does not obviously depend on knowing that everything will eventually end. Third, there is something troubling about the move of finding consolation in death. Death is, by any reasonable measure, bad. It deprives people of future experiences, severs relationships, and causes grief. The philosophical work of finding meaning in mortality can be seen as a coping mechanism for something genuinely terrible, rather than evidence that death is actually good. As Aubrey de Grey likes to point out, people did not argue that we should not cure smallpox because an early death from smallpox gave life meaning. A different category of objection concerns what immortality would do to society. The most frequently voiced concern is overpopulation. If people stop dying but continue to be born, the population would grow indefinitely. Earth's resources are finite. The math seems simple and catastrophic. But this objection, while intuitive, is actually more complicated than it appears. Fertility rates have been declining globally for decades, and in many developed countries are already below replacement level. If people lived indefinitely, it seems likely that the social and economic calculus around having children would shift further. People might choose to have fewer or no children, knowing that they would be around indefinitely themselves. Some futurists speculate that an immortal society might actually have a lower rate of population growth than a mortal one. This is speculative, but the automatic assumption that immortality leads to exponential population growth is not supported by a careful analysis. Furthermore, humanity is already working on questions of resource scarcity independent of immortality. Renewable energy, vertical farming, lab-grown meat, colonization of other planets. These are all technologies being actively developed. The capacity of Earth to support a large population is not a fixed number. It depends on technology. An immortal humanity would have far more time to develop the technologies needed to expand its resource base. Then there is the inequality objection. Longevity treatments will presumably be expensive, at least initially. In a world where immortality exists but is only accessible to the wealthy, you would create a society stratified not just by income and opportunity, but by lifespan itself. The billionaires live forever. Everyone else gets the normal allocation of decades. This is a genuinely alarming prospect. It is worth noting, however, that this argument proves too much. By the same logic, we should not develop cancer treatments, heart treatments, or any other expensive medical therapies, because initially they are only available to the wealthy. Almost every medical advance in history started out expensive and gradually became cheaper and more widely available. Statins, which dramatically reduce the risk of heart attack, were initially expensive and now are inexpensive generic drugs available even in low-income countries. The same trajectory could and should be pursued for longevity treatments. The existence of a problem with initial distribution does not mean the underlying technology should not be developed, but this requires active political and social work. Longevity science should not be developed only in the private sector, driven by the priorities of billionaires who want to live forever. It should be developed with explicit attention to global access and equity. This is a challenge that applies to all medical technology, but is particularly acute for something as fundamental as life and death. A deeper philosophical objection to immortality concerns identity over time. If you live for thousands of years, accumulating experiences, updating your values, changing your interests, growing in ways that cannot be foreseen, are you still the same person who was born in the 21st century? The ship of Theseus problem applied to the self. If every plank of the ship is eventually replaced, is it still the same ship? If every belief, every memory, every personality trait eventually changes, is it still you who is immortal? This is a genuine philosophical puzzle, but it is not actually specific to immortality. We already change substantially over the course of a human lifetime. The person you are at 70 shares few beliefs, memories, or personality traits with the person you were at 7. We accept this as the same person through a continuous chain of psychological and physical connection, not through the preservation of some unchanging essence. There is no obvious reason why extending this chain of continuity over thousands of years would break something that is not already being strained over a normal human lifetime. What about the psychological experience of extreme longevity? Would people become bored, depressed? Would they run out of things to want or to feel? This is an empirical question that we cannot answer with certainty because we have no data on what it is like to live for centuries. But human beings are extraordinarily adaptable, and the universe is extraordinarily large and complex. The possibility of existential boredom on a cosmic timescale seems less immediately pressing than the possibility of dying of cancer or dementia within the next few decades. There is also the question of how society changes when its members are immortal. Politics, culture, and institutions are in large part shaped by the regular turnover of generations. New people bring new ideas, new values, new energy. Old power structures die with the people who hold them. If people lived indefinitely, would entrenched power become even more entrenched? Would the dead hand of previous generations lie even more heavily on the present? These are legitimate concerns, but they are also somewhat speculative. An immortal person is not a static person. People continue to learn, change, and update over their lifetimes. The assumption that an immortal oligarch would maintain exactly the same values and power structures for millennia seems unrealistic. Though the risk of concentration of power in a world with radical life extension is real and worth taking seriously. What about the relationship between immortality and religion? For billions of people, the belief in an afterlife is central to their worldview and their moral framework. The promise of heaven, the threat of hell, the concept of karma and rebirth, these beliefs are shaped by and in turn shape how people think about death. If death could be abolished, would this undermine these belief systems? Would it create a crisis of meaning for people whose entire moral framework is organized around the transition to an afterlife? This is a real social and psychological challenge, and the cultural adjustments required would be profound. But religions have always adapted to new realities. The introduction of modern medicine, which dramatically reduced mortality from many causes, did not destroy religion. It changed how religions interpreted the relationship between the divine will and human health. A similar adaptation could occur in a world with radical life extension. And for secular people, and for religious traditions that emphasize life in this world, there are many. The prospect of a longer life simply means more life, more opportunity for meaning, love, and growth. Let's come back to the science, because there is an important question we have not yet addressed. How long do we actually have? The honest answer is that nobody knows. The uncertainty in predictions about longevity science is enormous. but we can identify some markers of progress that will tell us how close we are. The near-term picture over the next 5 to 15 years looks like this. Senolytic drugs are likely to receive FDA approval for at least one indication, possibly inflammatory conditions, pulmonary fibrosis, or musculoskeletal disease, within the next few years, based on the current trajectory of clinical trials. This would not be approval for life extension per se, but it would be approval for drugs targeting one of the fundamental mechanisms of aging. The TAME trial with metformin should produce results within the next few years. Partial epigenetic reprogramming will be tested in humans for specific diseases, probably ophthalmological conditions, since the eye is relatively accessible, and David Sinclair's lab has already shown impressive results there. The biological age clocks will improve significantly, providing better tools for measuring the efficacy of longevity interventions in clinical trials. AI-assisted drug discovery will accelerate the identification of novel longevity compounds. In the medium term, over the next 15 to 30 years, if the near-term progress goes well, we might see approved therapies that meaningfully slow aging in humans, early gene therapy approaches targeting aging-associated genetic factors, increasingly sophisticated epigenetic reprogramming therapies applied to multiple tissues, and possibly early nanotechnology-based drug delivery systems that can perform targeted cellular repair. The long-term picture, beyond 30 years, is where the uncertainty becomes very large. This is the territory of Kurzweil's predictions and de Grey's sense roadmap. It is possible that within 50 years, the combination of biological research and AI will have produced interventions that can maintain biological youth indefinitely. It is also possible that the translation from mouse models to humans will prove far more difficult than expected, that unforeseen complications will arise, and that meaningful life extension will remain elusive for much longer. What can we say with confidence? Several things. First, the science of aging has genuinely and substantially advanced over the past 30 years. Things that were science fiction in the 1990s, partial epigenetic reprogramming of tissues, targeted senolytic drugs, AI-designed longevity compounds, are active areas of well-funded research today. Second, the pace of progress is accelerating due to the influx of investment and the application of AI tools. Third, the timeline from basic science to clinical application in humans has historically been long and unpredictable. The history of medicine is littered with promising mouse studies that failed to translate to humans. Fourth, the fundamental scientific case for the possibility of dramatically extended human lifespan is stronger today than at any point in history. The biological mechanisms of aging are understood at a level of detail that makes targeted intervention genuinely plausible. Perhaps the most important thing to understand is this: we are not trying to predict when immortality will be achieved, as though it were a single event like a moon landing. We are describing a landscape of research along a spectrum. At one end, you have things like improved diet and exercise advice that might add a few years of healthy life. At the other end, you have the full abolition of aging and the possibility of indefinite lifespan. Between these poles, there are 100 different kinds of interventions with 100 different potential impacts. Progress along this spectrum is already happening and will continue. The question is how far it will go and how fast. For people who are alive today, the most relevant question is probably a personal one. What should I do? The answer, interestingly, is both boring and revolutionary. The boring part: the lifestyle factors that are well established to extend healthy life, regular exercise, a healthy diet, adequate sleep, not smoking, limiting alcohol, maintaining social connections, remain the most powerful tools currently available. The science of aging has not yet produced interventions that reliably outperform these basic lifestyle factors for most people. If you want to maximize your chances of still being alive when the more advanced therapies become available, the best thing you can do is maintain your health using established means. The revolutionary part: if you have a physician who is knowledgeable about longevity medicine, there are already some interventions with reasonable evidence behind them. Rapamycin, a drug originally developed as an immunosuppressant, has been shown to extend the lifespan of mice more robustly than perhaps any other pharmaceutical intervention, and some longevity physicians are beginning to prescribe it off-label in humans at low doses. The NAD+ precursors NMN and NR are widely used by longevity-focused individuals, with some supportive evidence. Metformin is prescribed off-label by many longevity physicians. These are all areas of active debate, and none of them have been proven to extend human lifespan in clinical trials. But the risk profiles are generally low, and the potential benefits are substantial if the underlying biology translates from mice to humans. Beyond individual choices, the most important thing you can do is to understand the landscape and advocate for it. Advocate for more funding for aging research. Advocate for the regulatory frameworks that will be needed to test longevity interventions properly. Advocate for equity and access to longevity medicine. The future of human immortality is not something that will happen to us. It is something we will collectively decide to pursue, fund, research, regulate, and distribute. Now let's dwell a little longer on something that deserves more space than it usually gets in these discussions: the psychological reality of living an extraordinarily long life. Not the politics, not the biology, not the philosophy of identity, but the lived experience. What would it actually feel like to live for 500 years? For 1,000? We have very little data on this for obvious reasons, but we can reason carefully about it from what we know about human psychology. Human beings are extraordinarily adaptable. The experience of living through a major historical shift, a war, a revolution, the invention of the internet, already stretches across a normal lifetime. People who were born before commercial aviation existed and lived to see the moon landing had to remake their understanding of the possible multiple times within a single life. People alive today have witnessed the complete transformation of how human beings communicate, find information, and understand themselves, all within a few decades. The human mind is capable of absorbing enormous amounts of change. An immortal person living across centuries would experience this kind of upheaval not once or twice, but dozens of times. The world they were born into would become unrecognizable. The language would change, the values would shift, the technology would transform the conditions of daily life beyond imagination. Is this exciting or terrifying? The honest answer is probably both. Human beings are deeply social creatures who build their sense of identity partly through shared cultural reference. An immortal person who outlived the cultural context in which they formed their identity would face a unique psychological challenge: the need to continuously remake their sense of self without losing the thread of who they are. This is not obviously impossible. People already navigate major identity transformations: religious conversion, emigration to a new country, the death of everyone one grew up with. These experiences are painful, but people emerge from them. They find new communities, new frames of reference, new sources of meaning. An immortal person would simply face this challenge on a grander scale and more frequently. There is also the question of memory. The human brain does not have unlimited storage. We forget the vast majority of what we experience. Over a normal human lifetime, the brain manages this through a combination of selective retention and fading. Over hundreds of years, the challenge would be different. Would an immortal person remember their childhood as vividly as their most recent decade? Almost certainly not. Would they carry meaningful emotional connections to events that happened centuries ago? The research on human memory suggests that emotional salience is the primary determinant of what we retain. Events that matter deeply, formative relationships, major losses, peak experiences, tend to persist far longer than routine events. This suggests that an immortal person would have a rich reservoir of highly meaningful memories, while the routine fabric of distant decades faded into something more like a general sense of accumulated experience. The grief question is particularly poignant. In a world where radical life extension is imperfect or unequally distributed, or where accidents and violence remain possible even if aging does not kill, immortal people would still lose people they love. They would watch friends and loved ones die from the causes that remain. Accidents, homicide, suicide, rare diseases that have not yet been cured. For a person who has lived for centuries and formed deep bonds with many people, the accumulated grief of losing them would be formidable. This is not a reason against immortality. After all, mortals already face the loss of everyone they love in the normal course of their lives. But it is a dimension of the psychological landscape of immortality that tends to be underexamined. Then there is the question of boredom, not in the trivial sense of having nothing to do on a rainy afternoon, but in the deeper sense of existential satiation. Could a person exhaust the meaningful possibilities of human experience? Could they reach a point where nothing was new, nothing was surprising, nothing was able to engage their attention and care? This is the intuition behind Bernard Williams's argument, and it deserves respect even if ultimately rejected. The counterargument is that the universe is genuinely inexhaustible. The number of books that could be written, the number of pieces of music that could be composed, the number of scientific questions that could be investigated, the number of relationships that could be formed, these are not finite. The space of human experience is incomprehensibly vast. If a person's curiosity and openness remain intact, there seems to be no obvious horizon at which the world would be exhausted as a source of meaning and interest. The risk of boredom may be real, but it is a risk that depends heavily on the psychological character of the immortal person, and on whether the treatments that extend life also preserve the cognitive flexibility, emotional responsiveness, and capacity for wonder that make life rich. There is also something to be said about the potential contributions that long-lived individuals could make. We talk often about geniuses who died too young, Ramanujan, Mozart, Keats, Galois, without fully computing the loss. But even those who lived to what we consider normal old ages, Newton, Darwin, Einstein, spent significant portions of their most productive years in declining health and cognitive capacity. What if Newton had had another 50 years of vigorous mental life? What would Darwin have discovered with 2 additional healthy decades after the origin of species? The compounding of knowledge in a single mind over centuries would produce individuals whose understanding of complex domains was simply beyond what is possible in a normal lifetime. This could represent an extraordinary enrichment of human civilization. Let's take a step back and try to hold the full picture in view. We began with a simple question: can humanity live forever? And we have traced the answer through thousands of years of human longing, through the biology of aging, through the experiments of modern science, through the investments of billionaires and governments, through the potential of AI and gene editing and nanotechnology, through the philosophical and ethical challenges that immortality raises. Here is where we stand. Death from aging is not inevitable in the way that the laws of thermodynamics are inevitable. It is the result of specific biological processes that can, in principle, be understood and addressed. The understanding has advanced enormously. The tools to do something about it are developing rapidly. The investment and attention from both the scientific community and the broader world have never been greater. At the same time, we are very far from having solved the problem. The translation from mouse experiments to human therapies is notoriously difficult. The biology of human aging is more complex than any other organism we study. The regulatory, ethical, and social frameworks for handling radical life extension do not exist yet. And the philosophical questions about what it means to be an immortal being in a mortal world, or in a world that is transitioning from mortal to immortal, are profound and unresolved. What is genuinely new about this moment in history is that these questions are no longer purely speculative. They are practical. Scientists and physicians are developing longevity treatments. Biotech companies are running clinical trials. Regulatory bodies are beginning to grapple with how to handle aging as a medical target. And for the first time in the history of humanity, there are serious, credentialed scientists who believe that the people alive today might live to see the defeat of aging. Whether they are right is something only time will reveal. But the conversation has changed irrevocably. The question is no longer whether aging can be studied. It can. The question is no longer whether the biological mechanisms of aging are understood. They largely are. The question is whether the gap between our current understanding and a working medical intervention can be crossed, and how fast. History suggests that once science has a clear understanding of a problem, and the tools to address it, progress can be faster than anyone expected. The development of vaccines for COVID-19 in under a year. The development of effective HIV treatments that transformed AIDS from a death sentence to a manageable chronic condition within 2 decades of the virus being identified. The development of the first cancer immunotherapies that have put some previously terminal cancers into complete remission. Science can move with startling speed when resources, talent, and motivation align. Those resources, that talent, and that motivation are aligning around aging right now in a way that has no precedent in history. But let's not end on a purely optimistic note, because the full truth demands something more complicated. There are real reasons to be cautious. Every decade, someone predicts that radical life extension is just around the corner, and every decade the promised therapies fail to materialize in the predicted form. The body is not simply a machine to be engineered. It is an evolved system of extraordinary complexity, with redundancies and interactions and feedbacks that we are still only beginning to understand. Our ability to intervene in that system is real, but our ability to predict the consequences of those interventions is limited. History is full of medical interventions that seemed promising and later turned out to have unforeseen side effects, sometimes devastating ones. And even if the biology could be solved, the social and political dimensions of immortality are genuinely hard. Who gets access? At what price? What happens to democracy when some people have had centuries to accumulate wealth and power? What happens to innovation when the people who benefit from the current system never die? What happens to cultural vitality when no generation ever fully yields to the next? These are not merely theoretical questions. They are the questions that would determine whether a world with radical life extension would be a human paradise or something more troubling. These challenges are real. But here is the thing. They are not reasons to stop. They are reasons to be thoughtful, to invest not just in the biology but in the ethics, not just in the science but in the governance, not just in the extension of life but in the quality and equity of the extended lives. The story of humanity is, in one sense, the story of a species that refused to accept the limits that nature imposed on it. We refused to accept that we could not fly, and we built airplanes. We refused to accept that information could only travel at the speed of a human messenger, and we built the internet. We refused to accept that smallpox or polio were simply facts of life, and we eradicated them. We refused to accept that the distance between stars was an insurmountable barrier to exploration, and we have now sent spacecraft beyond our solar system. Each of these refusals was met with skepticism. Each was described as impossible, hubristic, or dangerous. Each turned out to be achievable, and each transformation has changed the human condition in ways that were impossible to fully anticipate in advance. The refusal to accept death may be the most ambitious refusal in the history of the species. It is also the most consequential. If it succeeds, even partially, it will change everything about what it means to be human. Consider for a moment what a world without aging would look like. Not the grim dystopia of overpopulation and resource exhaustion that pessimists predict, but the best version of such a world. A world where no one watches their parents fade away, where no one spends the last decades of their life in diminishing capacity and increasing pain, where the accumulated wisdom of a long life is not erased but deepened, where the greatest thinkers, artists, scientists, and teachers have not 70 or 80 years in which to do their work, but centuries, where the tragic loss of potential that happens every time a brilliant mind is cut short at 50 or 60 or 70 by age-related disease simply does not happen anymore. What would Einstein have discovered with 200 years to think? What would Beethoven have composed? What would Marie Curie have uncovered? These are unanswerable questions, but they gesture at something real. The cost of mortality is not just the loss of individual lives, but the loss of what those lives might have produced. Every generation of humanity is, in a sense, a fresh start, but also a loss, because so much accumulated knowledge, experience, and relationship dies with the generation that held it. There is a powerful moral case for the elimination of aging that is rarely made as explicitly as it deserves to be. Aging kills 100,000 people per day. Every day. Over 36 million people per year. These deaths are not gentle or pleasant. They are preceded in most cases by years or decades of increasing pain, disability, and loss of autonomy. Alzheimer's disease, which strips people of their memories and personality, and then their ability to swallow, affects over 50 million people globally. Cardiovascular disease, which kills more people than any other cause, is an aging disease. Cancer, which kills another 10 million people per year, is predominantly an aging disease. Osteoporosis, arthritis, macular degeneration, Parkinson's. The litany of age-related suffering is immense. If aging is a biological process rather than an inevitable fate, then failing to treat it is not a neutral choice. It is a choice to allow an enormous amount of suffering and death to continue when we might have the ability to prevent it. The moral weight of this is staggering, and it should be felt more acutely than it is. We have, to some extent, normalized aging in a way that makes the deaths it causes feel less urgent than other deaths. We are horrified by a child dying of cancer, and rightly so. We are moved by a young person dying of AIDS or in a war. But when an 80-year-old dies of heart failure after years of cognitive decline, we say they had a good long life, as if that makes the death acceptable. They were a person. They had people who loved them. They had more they might have done, more they might have experienced, more they might have been. The loss is real, even if we have become accustomed to it. The goal of longevity science is, at its deepest level, to eliminate this normalized suffering. To say that it is not acceptable for human beings to decline and die from aging just because it has always happened. To say that if we have the intelligence to understand the mechanisms of aging and the tools to address them, then we have an obligation to do so. Whether we succeed in this generation or the next or many generations from now is uncertain. But the great project of understanding and defeating aging, is one of the most important things humanity has ever undertaken. It deserves more attention, more resources, and more moral seriousness than it has received. Let's spend a moment on something that rarely gets discussed in these conversations. The people who are already living as though immortality is coming, not as a thought experiment, but as a practical orientation to their own biology and their own futures. Brian Johnson is perhaps the most publicly visible example. A technology entrepreneur who sold his company Braintree to PayPal for $800 million. Johnson now spends somewhere in the vicinity of $2 million a year on what he calls Project Blueprint, an obsessively documented attempt to reverse his biological age through a combination of optimized nutrition, sleep protocols, targeted supplementation, regular medical testing, and an assortment of experimental interventions including plasma transfusions and various pharmaceutical compounds. He has subjected himself to hundreds of blood tests, continuous glucose monitoring, nightly sleep tracking, and detailed imaging of every organ system. His stated goal is to have the biological metrics of an 18-year-old across every measurable parameter. The project is frequently mocked, and some of the mockery is fair, because Johnson sometimes adopts a kind of performative extremism that invites ridicule. But beneath the spectacle is something genuine, a rigorous, data-driven attempt to operationalize the science of longevity in a single human body in real time. Johnson's most recent biological age measurements, using some of the best available clocks, suggest that his biological age is indeed significantly younger than his chronological age, though the interpretation of these measurements is contested. More interesting than the specific numbers is the approach, the idea that aging can and should be treated as a trackable, measurable, manageable process rather than a background condition of life that one accepts passively. Peter Attia, a physician and one of the most influential communicators in the longevity medicine space, takes a somewhat different approach. Less theatrical than Johnson, more clinically rigorous, Attia argues that the medicine of the future needs to be fundamentally reoriented. Rather than treating diseases after they have manifested, the reactive model that currently dominates medicine, longevity medicine should identify and address the precursors of age-related disease decades before they become symptomatic. He calls this medicine 3.0, a proactive, personalized approach to health that starts intervening on aging processes in people's 30s and 40s, rather than waiting for the heart attack or cancer diagnosis in their 60s and 70s. Attia's approach is grounded in the mainstream science but pushes at its frontier. He uses continuous glucose monitors, detailed cardiovascular imaging, comprehensive blood panels, genetic testing, and structured exercise and nutrition protocols to build a detailed picture of each patient's individual risk profile, and then intervene aggressively and early. The drugs and supplements he considers include rapamycin, metformin, and various others that remain in the territory between conventional medicine and experimental longevity intervention. What both Johnson and Attia illustrate, through their very different styles and methods, is that there is an emerging culture of longevity medicine that is willing to act on the current best science even before that science has been definitively proven in long-term human trials. This culture is controversial within mainstream medicine, where the standard of care requires much stronger evidence of efficacy and safety before recommending interventions. But it is also arguably a rational response to the available evidence and the very high stakes. If you wait for definitive proof of longevity interventions in humans, you may have to wait decades for results, by which point the opportunity for the people alive today may have passed. There is also an important collective dimension to this. The longevity advocates, the biohackers, the longevity physicians who are willing to prescribe rapamycin off-label, they are, in a sense, generating data. Each person who tracks their biomarkers carefully while taking a longevity intervention is, however imperfectly, contributing to the evidence base about what these interventions do in humans. This is not a substitute for randomized controlled trials, but it is a form of distributed experimentation that the traditional medical system does not and cannot conduct at this scale. The question of regulatory frameworks is crucial here. The FDA's current framework classifies aging as a natural process rather than a disease, which means that drugs cannot be approved with slowing aging as their indication. The TAME trial with metformin is attempting to change this precedent by using aging itself as the trial's endpoint, essentially arguing to regulators that aging is a modifiable condition and that drugs targeting it should be approvable. If TAME succeeds, it will open a pathway for approving future, more powerful longevity drugs in a way that the current framework does not allow. Some researchers and advocates have argued that the pace of longevity drug development should be dramatically accelerated through regulatory reform. They argue that the risk-benefit calculus for aging interventions with good safety profiles is fundamentally different from the calculus for drugs treating acute diseases. If a cancer drug causes serious side effects in some patients, those side effects are weighed against the benefit of treating an immediately life-threatening condition. If a longevity drug causes minor side effects in some users, those side effects are weighed against the potential benefit of extending healthy lifespan by years or decades. The calculation may justify a different threshold for approval, particularly for interventions with well-characterized safety profiles. This regulatory conversation is happening, slowly, in governments and health agencies around the world. The UK's Medicines and Healthcare Products Regulatory Agency has expressed openness to new frameworks for evaluating aging-related therapies. The FDA has initiated conversations with the aging research community about how to design trials for longevity interventions. These are early steps, but they represent movement in a direction that was unthinkable even a decade ago. There is also a growing movement of philanthropists, foundations, and government agencies that are directing significant resources specifically toward longevity science with the explicit aim of accelerating progress. The Longevity Vision Fund, the Life Extension Advocacy Foundation, the Methuselah Foundation, the Sense Research Foundation, Open Philanthropy. These are just a few of the organizations that have made defeating aging a priority. Combined with the billionaire-funded startups and the government programs mentioned earlier, the total capital flowing into longevity science is now larger than at any point in history. The momentum is real. Whether it will be sufficient and fast enough remains the great open question. So back to the question we started with. Can humanity live forever? The honest answer is, we don't know yet. But for the first time in the 4 billion years that life has existed on this planet, a species has developed the intelligence, the tools, and the will to seriously ask the question, and to begin doing the science that might answer it. We are at the beginning of something. Whether it is the beginning of the end of aging, or the beginning of a long road that will take many more generations to travel, we cannot say with certainty. What we can say is this: the question is real, the science is real, the stakes are as high as stakes can possibly be, and the possibility, not the certainty, but the genuine, scientifically grounded possibility, that people alive today might live to see the defeat of death itself, is not a fantasy. It is a hypothesis being tested in laboratories around the world. Right now, tonight, while you are watching this video, we are living at the most extraordinary inflection point in the history of the human relationship with death. Every prior generation faced aging without any realistic prospect of defeating it. They had no choice but to accept. They built philosophies of acceptance. They built religions of consolation. They passed on their wisdom and their love to the children who would replace them, and then they died. That is the human story from its beginning until now. A story of finite lives lived with courage, meaning, and resignation. What is different about this moment, what is genuinely, historically unprecedented, is that resignation is no longer clearly the only option. For the first time, scientists with the best tools available to humanity are actively working on the problem of aging and making genuine progress. They do not have the solution yet. They may not have it in our lifetimes, but the direction of travel is clear. Understanding is deepening. Technologies are maturing. The scientific consensus that aging can be addressed is firmer than it has ever been. We stand, perhaps, at something like the position of the Wright brothers in 1900. Human flight had been dreamed of for millennia. The physical principles that would make it possible were understood. The materials and engineering tools were just becoming adequate to the task. Most people still thought it was impossible, and within 4 years a machine left the ground and carried a human being through the air. Within 60 years we were on the moon. The defeat of aging may follow a similar arc. The dream is ancient. The scientific principles are being understood. The tools are maturing with extraordinary speed. Most people still think it is impossible. History suggests we should be cautious about that assumption. The last person who will die of aging may already have been born, or the first person who will live indefinitely may already be alive. Which one is true will depend in part on what we choose to do with the time we have. That is perhaps the most remarkable sentence that has ever been true. The end of death may be a matter of human choice. And that is where we are. If this video made you think about mortality differently, about what it means, what it costs, and what we might be able to do about it, then please share it. Not for the algorithm, because these ideas deserve to be part of the conversation.